As we move past Chapter 4 then, looking at cell structure, Chapter 5 is looking at the working cell. And there's really three major topics within this chapter that are important. One is the idea of, well, where did our eukaryotic cells come from? The second is cell transport. And the third is metabolism. And today we'll be looking at where our cells came from and cell transport. And as we mentioned last time with our cell structure, we said prokaryotic cells are very ancient. They've been around for about 3.5 billion years. Eukaryotic cells are also very old. They're about 2 billion years old. But that means for about 1.5 billion years, prokaryotic cells were the only form of life on our planet. And then we see eukaryotic cells appear in the fossil record. And so that certainly begs the question, where did those eukaryotic cells come from if it was only prokaryotic cells for such a very long time? And we explain that by a process called endosymbiosis. And endosymbiosis is a theory that explains the origin of our eukaryotic cells, our cells that are larger, more complex, a lot more of those cell parts. And not surprisingly, it involves those existing prokaryotic cells. So the prokaryotic cells that were already present, some of those evolved into our eukaryotic cells. And if we break down the word here, endosymbiosis, endo just means when one organism lives inside of another. And so, for example, if you had an endoparasite, that would be something like a tapeworm living inside, versus an ectoparasite would be something on the outside, like a tick or something like that. So endo, one organism is going to live inside another, in this case, one cell living inside of another cell. Symbiosis has to do with relationship, and there's lots of different relationships organisms can have. If they have a symbiotic relationship, that means the two organisms have a very close, tight relationship such that they both depend upon each other, and one organism could not live without the other. And so as we think about cells here, that means two cells are going to live together, one inside the other with that endo prefix, and both of them are going to benefit from this relationship. It's a mutualistic or a win-win type of relationship. They're both providing something that the other needs. Now, this process of endosymbiosis, one cell living inside another and both cells benefiting, is relatively common among protists today. In fact, we see this in certain types of free-swimming protists, especially in pond water, for example, where one cell will eat another cell, but instead of digesting and killing that cell inside, it will feed that cell inside, it will keep it alive. And the idea behind our theory of endosymbiosis is that cell that's kind of the host that's keeping the other cell alive. When it divides, those other cells inside of it are going to stay inside of it as it's passed from generation to generation, and those cells on the inside that have been eaten are gradually going to evolve and change and adjust to that internal environment and eventually evolved into those different cell organelles that we talked about last chapter. So if we take a look at a video here, this is looking at a eukaryotic protist, and we can see it's this large cell with these cilia that allow it to swim around, and it's full of these little green dots. Well, these little green dots are actually photosynthetic bacteria. These are cells that have been eaten. The host is keeping them alive. The bacteria inside get the benefit of having a safe place with lots of nutrients and sunlight. The host cell gets the benefit of having a food source, and so the bacteria produce a little extra food and they leak it into the cytoplasm and feed the host. So again, it's a win-win situation for both of these organisms. So a good example of endosymbiosis. So if we look at some lines of evidence, why do people think that this endosymbiosis may have led to eukaryotic cells? This is really imperative when we look at the energy organelles, those mitochondria and those chloroplasts that we talked about last time. Remember, these are two of our larger organelles, the mitochondria generating usable energy for your cell, the chloroplasts generating sugar from solar energy in plant cells. And the first clue was that these mitochondria and chloroplasts are the same size as free-swimming bacteria. They look a lot like prokaryotic cells. In addition, we know that most of our DNA is in the nucleus, but we also have some DNA in our mitochondria, and plants also have DNA in their chloroplast. And so these organelles not only are the same size as bacteria, but they actually have DNA. Not only that, but they have DNA that's in a circular form, which is very similar to that we find in free-swimming bacteria. And in fact, people have since sequenced the DNA, and we can tell that they're very closely related to certain branches or types of bacteria. Ribosomes, or remember, are those organelles that build proteins. And there's two types. There's eukaryotic ribosomes, and then prokaryotic ribosomes are a little bit different in structure. And mitochondria and chloroplasts not only have ribosomes like cells do, but they have the bacterial type of ribosome. And finally, both these organelles don't just have one wrapping of membrane or one phospholipid bilayer, but they have a double membrane. And that's evidence of something being eaten, because a cell typically would have its own single membrane, and then when a host eats it, it's going to wrap it in a second membrane. And so again, all these are strong evidence that these mitochondria and chloroplasts were once free-swimming bacteria that have evolved into these organelles. And if we take a look at a little clip here we're looking inside of a cell, and all these little dots moving are mitochondria, the tiny little dots. And again, they look just like a bacteria cell does under the microscope. Here we just have an animation. Here we can see the double membrane, the outside kind of oval membrane, and then that kind of convoluted or folded inner membrane. And again, that's evidence of something that's been eaten. And here in the clip we'll see a cell that's eaten has its own membrane, and then the cell that's the host doing the eating wraps it in that second layer or that second membrane. So again, strong evidence of a cell that was once free swimming. And our theory of endosymbiosis really only applies to the chloroplast and mitochondria as being free swimming. The other organelles or structures we see in the eukaryotic cells are thought to be due to foldings of the membrane. So, for example, the DNA in its own nuclear compartment, or that whole endomembrane system with the ER and the Golgi and the vacuoles and so on. It's thought that the membranes on the outside of the cell started to fold and mix inside the cell and within the cytoplasm, and that the nuclear membrane is going to provide protection for DNA from all those chemical reactions associated with things like mitochondria that had moved into the eukaryotic cell. And also, as we talked about last chapter, they compartmentalize or break up the cell into lots of small compartments. So here we have our endosymbiosis as a whole. It's all mitochondria. Those bacteria were the first taken in and involved with the mitochondria. Once that happened, the membrane folding started to close in the nucleus and form the endomembrane system. And that plant cells had a second round of endosymbiosis where they took in a photosynthetic cell, and that eventually became a chloroplast. So that's the idea of endosymbiosis, one of our major topics today. The other major topic is cell transport. How do cells get food in? Do they get rid of waste and kind of exchange materials? And so just as a reminder, last chapter we talked about the fluid mosaic model, that our membrane is made up of that phospholipid or fatty bilayer with lots of proteins dispersed here. Okay? And so we've got this very fluid, three-dimensional grouping of these phospholipids that allow certain chemicals to pass through. Other chemicals can't. And remember, we said that was being selectively permeable. And so we're going to take a look at how chemicals are going to cross this phospholipid bilayer. If we think big picture first, there's two major mechanisms or ways chemicals can get into or out of cells. The first is that most chemicals are going to pass through that membrane. So they're going to have to either go through the fatty phospholipid bilayer, or they're going to need the help of one of those transport proteins to get into or out of cells. And that's pretty intuitive. If this membrane wraps around the whole cell, things have to cross it to get in and out. But sometimes cells can actually kind of play a little cellular trick here, a little cellular game, and some chemicals can actually get into the cell or leave the cell without passing through the membrane at all. And I know that sounds counterintuitive. If the membrane's around the cell, how can something get in without getting across it? And we'll take a look at those. But certainly most chemicals get in by crossing the membrane, so we'll start off with part A there and then wrap up by looking at part B. And there are four major mechanisms that chemicals can use to cross a membrane, and it really doesn't matter whether it's going in or out. It's just a matter of getting across that membrane. And these are things you may be familiar with, things like diffusion, osmosis, facilitated diffusion, and then active transport, the four basic mechanisms here. And so we'll start with diffusion and just talk a little bit about each of these mechanisms, how chemicals get across, and then what are some examples of some chemicals that move by these different mechanisms. Diffusion is a good place to start because it's the most basic of all these movements of chemicals. And here's our official definition of diffusion. The idea that chemicals move from areas of high concentration to areas of lower concentration. Okay? Chemicals move from areas of high to areas of lower concentration. And so this is a basic mechanism or force of nature. This isn't something that cells do or that membranes have to actively do, but chemicals are going to gradually spread out. And that's, again, a sort of a natural movement. If I were to take, for example, some food coloring and drop it into a glass of water, the food coloring doesn't just stay in a little drop, but gradually it spreads out. And here's an example of why, and this is looking at an example with the membrane. Remember, chemicals are in constant motion. They're always moving around. And so if I have a bunch of chemical dyes on one side of a membrane, they're going to randomly start bouncing around. And some of them are going to go up and down, some are going to bounce into each other, and gradually, because they're always bouncing around, they're going to gradually tend to spread out over time. And so we're going to eventually, moving from right to left, end up in that equilibrium state there where we've got about an equal amount of the dye on both sides. And that's our natural process of diffusion. And diffusion is a natural force, just like gravity is a natural force, and so it's occurring out there in nature whether cells are around or not. But if cells are around, we think of this diffusion again as a natural flow. Cells don't have to spend any energy. Again, just think about if you're riding a bike and you're going downhill, you can coast. You don't have to spend energy because gravity or the earth is kind of doing the work. The same thing here. Chemicals are going to move by diffusion. The cell doesn't have to do anything. And so because of that, your book refers to diffusion as a passive process because, again, the cell's almost like a bystander. It's not actively participating here. And chemicals in diffusion are going to move from areas of high to areas of lower concentration. Again, they're going to spread out. And when chemicals move from high to low, we say that's moving with the concentration gradient. The chemicals are moving with that natural flow or with the gradient. And chemicals that diffuse into or out of cells tend to be small chemicals and very important chemicals like water, oxygen, and carbon dioxide. Those are all examples of things that will simply diffuse into or out of a cell. Now the direction, whether it's going to enter or exit the cell, is completely dependent upon where the concentrations are. It's always going to move from high to low. And so if there's a lot of oxygen, for example, on the outside of a cell, it will diffuse into the cell. Okay? And this is the major mechanism for these oxygen and carbon dioxide in our blood flow. And so let's take a look here at this diagram. If we think of a capillary, for example, in your lungs, when blood cells get to your lungs, they're low on oxygen, high on carbon dioxide. And so when you breathe in air, that air is high in oxygen and low in carbon dioxide. And so oxygen is going to diffuse from high to low, so it's going to move from the air into the blood cells. And so your blood cells will kind of get recharged or refilled up with oxygen by diffusion. Likewise, carbon dioxide levels or waste gases in your blood are going to be very high, and that carbon dioxide will diffuse out of your blood and into your lungs. Okay? So now your blood cells have lots of oxygen, very little carbon dioxide. They're going to get pumped back to the heart, pumped out again, and go to some cells in your body. And that's what they're showing here in this picture. And when your cells arrive, let's say in your little toe on your foot, they're going to be high in oxygen, and so that oxygen will spread out or diffuse out of the blood and into the cells, which are needing oxygen. Likewise, the cells have accumulated a lot of carbon dioxide because that's the waste product they're generating. And so that high carbon dioxide concentration in your cells is going to diffuse back into your blood. And then your blood cells are going to circulate, and the process is going to repeat itself. And so certainly our gas movement and circulation are directly tied to diffusion. And here's just an animation looking at diffusion. Again, chemicals gradually spread out, and so they're going to go from high to low. They're moving with that concentration gradient. And it doesn't really matter, again, whether a cell is there or not. If it's a chemical that's permeable, that can pass through the fatty layer, it's simply going to slide right through that fatty layer regardless of which chemical. And again, because water and oxygen and carbon dioxide tend to pass through the fatty layer, they're just going to diffuse into and out of cells. And again, it's very passive. The cell doesn't have to physically do anything to make this happen. Next up is osmosis. And osmosis is just a special case of diffusion. And so it's still diffusion. And so all of our diffusion rules are certainly going to apply here. But in particular, osmosis is the diffusion of water across a selectively permeable membrane. Okay? And so because water is so abundant in cells and the movement of water is so important to cells and cell metabolism, we just have a special category of osmosis of what happens when water diffuses into or out of a cell. And again, it's still diffusion, so all the rules that we just talked about apply. Osmosis is going to be passive. Water is going to move with the gradient from high concentrations of water to less water, and it's going to cross the membrane. So let's take a look at some issues here of osmosis, and then we'll look at a few examples here. Again, it's diffusion, so chemicals move from high to low concentration. So water is moving from areas of high concentration to low concentration. But here's one of the problems that makes osmosis, I think, confusing sometimes, is that because water is so abundant, we rarely think of the water or the water concentrations. For example, if you walk into a lab, you're never going to see a bottle that says 80% water or 10% water or 50% water. We never label the water because it's so common. Instead, you're probably going to see a bottle that says 10% glucose or 8% salt. And so typically we label the solute, the chemical we mix with water, and we don't label the water. Okay? But of course, if I've got a bottle that says 10% glucose, we always assume, well, the other 90% must be water. But again, we rarely label it, we rarely think about it. And in fact, we've got some terms here that describe concentrations, but those terms we use to think about concentrations refer to concentrations of solute, because again, that's usually what people are interested in. And so we're going to use these terms, but we have to remember we're interested in water, not the solute. So we have to take that extra step of going from sugar concentration or salt concentration and realizing that everything left over is water. And so let's kind of start thinking about water concentration, since that's important here in osmosis, and look at a couple of examples. For our example then, let's consider a beaker of water. And so here we have a beaker of water, and we're going to split the beaker into two halves using a membrane. And something like dialysis tubing is a good mimic for a plasma membrane. So we could just put some dialysis tubing and split my beaker in half there into side A and side B. And let's put 10% solute, let's just say salt, 10% salt on side A, and we'll put the same thing on side B, 10% salt. Well, if I've got the same concentration of solute or salt on both sides, that's called an isotonic solution. Okay? So both sides, in this case A and B, we would say are isotonic, meaning they have equal concentrations of solute. Okay? Now, again, we're interested in the water here, though, so we have to take the next step, and we're going to convert that then into obviously 90% water on side A and 90% water on side B. And so an isotonic situation technically refers to having equal concentrations of solute, but by default, we're also going to have equal concentrations of water. And so if we think of osmosis, we said osmosis moves from high concentration of water to low. In this case, I don't have a high or low. Both water concentrations are already equal. So in the isotonic example, we're not going to see any net movement of water. We won't see osmosis occurring because we're already at the equilibrium state. So one possibility is if we have isotonic, both sides are equal. Obviously, the only other possibility is what if both sides are not equal. So let's take a look at that example. So here's our beaker again. We'll split it in half with our membrane into two different sides. This time on side A, let's put 8% salt. And on side B, we'll put a different amount, let's say 5% solute or 5% salt. So in this case, side A has more salt than side B. And so we're going to use a pair of terms to describe these concentrations. Hypertonic refers to the side that has the higher solute concentration. And so in this case, side A is hypertonic. There's more solute, there's more salt. Side B then by default is less solute, and that's referred to as hypotonic. So the hypotonic is the lower solute concentration. Okay? And I know these terms, hypertonic and hypotonic, they sound a lot alike. They're easy to get mixed up. My little pneumatic kind of memory aid here to remember these is when I think of hypertonic, I think of hyperactive little kid. I've got 3 little kids at home. You give them sugar, too much energy, hyper. Right? And so think of hyper and more, too much. In this case, too much solute, higher solute concentrations. On the other side, hypotonic, think of that O and hypo and low. Hypo and low, if you are hypoglycemic, for example, you have low blood sugar. Okay? So in this case, that's the solute concentrations, hyper on one side and hypo on the other, but we have to take the next step and convert them into water concentrations. So on side A, I would have 92% water. And then on side B, obviously, I'd have 95% water to get me up to 100% on side B. And so in this case, water moves from high to low concentrations. Water is higher on side B, so it's going to move from side B towards side A. And so we would expect osmosis to occur toward side A. Okay? And in fact, water is always going to move from the hypotonic side toward the hypertonic side. That's a very predictable thing to remember here. Water is always going to move from the side that's hypotonic toward the side that's hypertonic. Okay? Now we may also ask the question, well, what about the solute? Shouldn't the solute diffuse from side A to side B? Shouldn't it spread out too? And the answer is yes, it should, that these chemicals tend to spread out. That's our natural movement. But remember, our plasma membrane is selectively permeable, and most solutes are chemicals like salts and sugars cannot cross the membrane. And so while there might be a tendency to spread out, that membrane is going to be a barrier and prevent the solute from moving. And so we're only going to see water moving in osmosis. Now if we apply our osmosis from our beakers actually to cells, here we can see some different solutions. And again, your body maintains an isotonic solution. So for example, your blood cells are normally kind of nice and round and donut shaped, and your blood plasma is isotonic, meaning your cells are not going to gain or lose water. That's part of our homeostasis, maintaining our consistent internal environment. But if we were to take your red blood cells, for example, and drop them into a hypotonic solution, okay, and if the solution is hypotonic, that means low sugar, okay, low solute, that means inside the cell would be hypertonic. And we said water always moves from hypo toward hyper. And so in this case, water would move into the blood cell. This blood cell would actually swell up and it actually burst or lyse, kind of like a water balloon, and that eventually would kill the cell. If we look at the last example, if we drop your cell into a high salt solution, a hypertonic solution, that means it's hypo inside, hyper outside. And so the water is going to leave the cell, and the cell will actually, what's called crenation, it'll actually shrivel up and look very raisiny and crinkled. Okay? And water will move in these directions for animal and plant cells the same way. Okay, so osmosis is going to affect both cells similar. The only difference, though, with the plant cells is plant cells, remember, have a big, thick plant cell wall, and so that plant cell is not going to swell up and burst like an animal cell. It may swell up and be under a lot of pressure, but you can't visually see that water pressure because that cell is going to be held together by the cell wall. So here we're looking at a bunch of plant cells. Each plant cell has a big, thick cell wall, that little brick, and lots of green chloroplasts. And this plant cell has been put on high salt solution in a hypertonic solution, and we're looking at time lapse. And notice inside each cell, the cells are shrinking. They're losing water here, and we can see the cell is getting smaller and smaller. Again, the cell wall isn't changing in shape. The little brick structure is staying the same, but the cell inside is gradually shrinking and getting smaller, and that's due to that osmosis in the high salt solution. Our next mechanism of transport is facilitated diffusion. And in facilitated diffusion, that's the diffusion across the membrane using the help of one of those transport proteins. And so it's still going to be diffusion, chemicals moving from areas of high to low concentration, but these are going to be chemicals that cannot cross through the phospholipid bilayer, that fatty layer, that they're going to need the help of one of these transport proteins. And that includes two groups of chemicals that need help across the membrane. Chemicals that are too large to fit. For example, things like sugars or proteins can't fit through that fatty layer, or chemicals that have a really strong charge, ions like sodium or chlorine, for example, need to move through the membrane through facilitated diffusion. Now we're still talking about diffusion here, so all of our diffusion rules that we've talked about still apply, so nothing has really changed here. Chemicals are moving from high to low or with the gradient. It's still a passive transport. Cells don't have to spend any energy here. This is just happening. And so in the picture here we can see a simple transport protein makes a little tunnel in the membrane. Small chemicals like ions can just simply pass in and out, and they're going to move from high to low, that natural movement of chemicals. The last mechanism to cross the membrane is active transport. And active transport is different from the other three because it is going to be active, as the name implies here. And in this case, chemicals are going to be moved from areas of low concentration to areas of higher concentration. Okay? And so sometimes cells need to take chemicals that are very rare and concentrate them where the cell needs a lot of that particular chemical, and that's going to happen here in active transport. Now this is going against the concentration gradient. It's going against the natural flow because chemicals tend to spread out, and here we're going to try to concentrate them. We're going to try to put all of that chemical together. And the analogy here is if you're going uphill on your bike, right? If you're going downhill, we said that's passive. Gravity does the work. If you're going uphill, though, you're going against that force. You have to add energy. The exact same thing applies here. In active transport, we're kind of going uphill. We're going against that concentration gradient, and so cells have to spend energy to make this happen. Okay? In addition to that, cells also need those transport proteins, and so it's a combination of energy and transport proteins to work as pumps that are going to allow active transport to occur in cells. And here's an example of an active transport protein. This is called the sodium-potassium pump, with the little purple protein in the middle, and this was one of the first active transport proteins ever discovered. And it can hold 3 sodium ions. It has to spend some energy, which is ATP, that works the pump and pumps the sodiums out, and that ATP has enough energy to also pump 2 potassiums in. And so this pump is kind of doing double duty. Sodiums get pumped out, potassiums get pumped in with one bundle of energy here. And again, notice we're going against the gradient. We're going from low to high. And so in this case, the sodium ions are being concentrated outside, the potassium inside. Okay? Now, we might think, well, shouldn't those ions undergo diffusion and spread out? And the answer is yes, they should. We might expect those sodium ions to come rushing back in. But remember, that phospholipid bilayer is a barrier. It prevents those ions from crossing. And so the only way sodium can cross is these transport proteins, and the same obviously for potassium. And so the cell can set up these concentration gradients using these active transport proteins. To finish up our transport across the membrane, I wanted to think about water regulation in particular, because as we mentioned, water simply moves by osmosis. Water moves from areas of high to areas of low water. It tends to spread out, and it can just leak into or out of cells. It can just come across that membrane any time. And so that begs the question then, how can cells control water movement if water is just leaking all over the place? Because cells certainly have to be able to do that. They have to regulate water and water movement. Well, it turns out water regulation is due to a combination of active transport and osmosis together. Okay? That cells cannot directly move water or pump water very effectively, but what they can do is they can alter concentrations to cause osmosis to occur. So, for example, if a cell had to move water, it would first start by active transport of solutes, typically salts, and it would pump salts across the membrane. And by pumping those salts, what that does is that changes the solute or the salt concentration, which we know indirectly is also going to change the water concentration. For example, if I add more salt to one side of the membrane, that side of the membrane now has a lower water concentration as well. Okay? And then water is going to move based on osmosis due to these new concentrations. And so again, the cell's not directly moving the water, but it can indirectly move it by changing solute concentrations, which then is going to cause osmosis to occur in different directions. So let's take a look at an example here of this indirect water movement. Let's say we've got a cell that needs to lose water. It needs to remove some water from inside the cell. The cell would start out then by pumping salts out. Using active transport and salt proteins or salt channels, it would have to spend energy to pump salts out to concentrate the salt. Okay? Once it did that, the water would then leave the cell following those salts due to osmosis. Okay? So for example, let's take a look at our situation here. Let's look at our cell. We've got inside the cell, there's our cell membrane, and outside the cell. And so on our original setting, let's say we've got 2% salt or solute on the inside and 1% solute on the outside. Okay? Which means inside we'd have 98% water, outside 99% water. And so here at the beginning, our water, remember, would osmose from high to low water, so we would expect water actually to be leaking into the cell, moving inside the cell by osmosis, which is the exact opposite direction the cell needs. Remember, the cell needs to pump out or lose water. So the cell's going to do that by move the water by pumping the solutes, and so active transport is going to occur to start changing the salt concentrations. So that if we came back to that cell in maybe 10 or 15 minutes, what we would see is that now most of the salt has been pumped out of that cell. So maybe it only has 1% solute left, and that other solute is already on the outside of the cell, which means my water percentages have changed, again, indirectly, and now water is going to move through osmosis from high to low, and so water is actually going to leave this cell. And so again, it's a very indirect way to move water, changing solute concentrations by active transport first, and then water is going to move by osmosis second. And this issue of water movement and water regulation is incredibly important, and one of our more common genetic disorders in humans is cystic fibrosis. Okay? And cystic fibrosis is a genetic disorder, so you cannot catch it, but you can inherit it from mom and dad. And cystic fibrosis' major issue is it's a problem with this water regulation. It's a problem with what we just looked at on the last slide, moving water out of the cell when you need to. Now, cystic fibrosis is a particularly hard disease to treat because it's a systemic disease, meaning it affects your whole body. It doesn't just affect one organ or your skin or your brain, but it affects your whole body. Especially it affects a lot of the respiratory system, the digestive system, and even the reproductive system. Okay? And it turns out that cystic fibrosis is due to a problem with one of these cell membrane proteins, in particular one of these salt channels and these salt pumps that we looked at. Okay? And here we can see in the picture a normal salt pump has this particular shape. Remember, it's a protein, it's a chain of amino acids, it folds up, it goes into the membrane, and it allows active transport of salt. And that means the cell can move the salt, and in turn, osmosis will occur. But if you have cystic fibrosis, you have a protein more similar to the one on the right. You have a mutation, one of your amino acids is different, that changes the shape of your protein, and the protein no longer fits into the membrane, and so it no longer works. So someone with cystic fibrosis cannot pump salt, which means they cannot regulate their water. And by far the most common symptom of cystic fibrosis is very thick mucus and bodily fluids, because they have no way to dilute their fluids or get water out of cells and into their fluids. So with the respiratory tract, for example, the mucus in the lungs is very thick. And so normally we all have watery mucus in our lungs, not too nice to think about, but that's the way it is. And if we have a cold or you're sick, you breathe in some contaminant, you cough and you sneeze and you hack and you get it out. But someone with cystic fibrosis, their mucus is so thick and heavy that it can't be moved up and out. And so the lungs being warm and wet and dark is a good place for bacteria to grow. And so a lot of pneumonia or other respiratory infections associated with cystic fibrosis. In terms of digestion, The digestive juices and fluids are very thick, so that inhibits normal digestion. And a lot of people with cystic fibrosis are sterile or have reproductive problems because those reproductive fluids are going to be very thick and mucousy as well. Okay. Now, today, certainly there is no cure. We can't go in and fix this problem, unfortunately. We know what the problem is. We can't fix it just yet. But there are lots of treatments. Chest massage is done, and that's just taking your child and patting their back very soundly 4 or 5 times, and you do that several times a day to try to break up the mucus in the lungs so that it's going to move. And here you can see in the picture, and it makes it look a lot worse than it really is there. For digestive problems, we can give you enzymes to take with your meals, and whereas cystic fibrosis at one time was very fatal to adolescents, today we see life expectancies gradually creeping up into the early to mid-30s. And so again, here's a very serious human disorder directly linked to this issue of water regulation and transport that we're talking about today. Now, if you remember way back when we were getting started today, we said there were 2 main transport issues, either crossing the membrane or not crossing. And so our second part here, part B, is transport without crossing the membrane. And we can split it up into whether the chemicals are entering or exiting the cell without crossing the membrane. And again, a little cellular trick here that cells can play. And so our first mechanism, exocytosis, is when chemicals are going to exit the cell. And so think exit exocytosis. So the cell's going to excrete or get rid of chemicals, and those chemicals are going to leave the cell, but not actually crossing the membrane. And so if we take a look at a picture here, cells oftentimes are going to pack chemicals inside a vacuole. And so we can see this little vacuole. It's basically a membrane sac, our phospholipid bilayer. And that vacuole is going to get pushed up to the edge of the cell using the cytoskeleton. Because the vacuole and the cell membrane are both made up of the phospholipids, those 2 things, when they bump into each other, are going to merge together. And then the cytoskeleton actually will act to pull and open up that vacuole. And once it opens up the vacuole, the contents inside are going to spill out of the cell. And so here our chemical, represented by our little green dots, started inside our cell. They ended up outside of the cell. But again, they never actually physically went through the membrane. Instead, the membrane was sort of moved out of the way for the cell. And the opposite of exocytosis then would be endocytosis, when chemicals are going to enter the cell. And again, think endo, enter, and they're not going to cross the membrane. And again, this is just basically just the opposite. If there's a lot of chemical outside that a cell needs to take in, it can form a little pit or dimple with the cytoskeleton. Once some of those chemicals bounce into the little dimple, then the cytoskeleton will pinch that dimple closed, and that chemical will be brought into the cell inside of a vacuole. And so again, the chemical goes from outside to in, never actually crosses the membrane. There are 3 different types of endocytosis, just depending on what's being brought into the cell. One common type is called phagocytosis. And phagocytosis is when cells are eating large food particles or even eating an entire new cell, another cell, as their food. And so they form this large pit. Once the food is inside, it's going to surround the food and pull it inside, and that food's going to end up inside of a vacuole, sort of like a little temporary stomach inside the cell. And this is commonly what we think of if we've seen a little clip of an amoeba, for example, kind of surrounding and eating another cell. Penocytosis is another type of endocytosis. Penocytosis is endocytosis of chemicals. Okay, so smaller things other than food. And so in this diagram, for example, we see the cell makes a little pit. Any chemicals that are bouncing around outside the cell are going to end up in the pit. It's going to pinch off the pit and move it inside. And actually we see under the electron microscope here, a picture on the right, here's a cell with several pits and undergoing lots of penocytosis. Now, one of the drawbacks, though, to penocytosis is what if the cell needs to take in a very specific chemical and there's not very much of it outside the cell. Then when the cell makes a pit and pinches it off and brings it inside, it may come up empty. And so a way to avoid coming up empty is this last type of endocytosis that's called receptor-mediated endocytosis. And this is very specialized. And cells have these receptor proteins that stick out of the membrane. And these receptor proteins will bind to the specific chemical needed. In the diagram, the little purple triangles there. And so the cell will make a pit, but it's not going to pinch off the pit and form the vacuole until all of the proteins are full. And so once the proteins have bound up the chemical, then it will make the pit and pull it inside. And that's a way of ensuring that that chemical is going to be captured and actually brought inside. And on the electron micrograph here on the right, we can see the line going through making the pit is a plasma membrane. We've got lots of proteins on the surface, and we can see a few of those proteins are full. They have the chemical actually attached to them. And it turns out that this receptor-mediated endocytosis is actually very important in us as humans, and if it doesn't work properly, is linked to lots of types of heart disease. Okay? And we mentioned HDLs and LDLs before as our different types of fats in our blood, and we said the LDLs were the bad fats. They're the fats that end up getting converted into cholesterol. Well, it turns out if you've got lots of LDLs in your blood, your body's going to try to remove them first before they're converted into cholesterol. And we remove these LDLs from our blood through receptor-mediated endocytosis, that our red blood cells have little protein receptors that bind these LDLs and take them inside. And if we do that, those LDLs get digested by your blood and they don't get converted into cholesterol. Well, it turns out that whether or not you have these protein receptors and how many receptors you have is a genetic trait that's inherited from your parents. And so if you don't have these receptors, that means the LDLs stay in your blood and they're going to get converted to cholesterol. So you're going to be much more likely to develop heart disease, hardening of the arteries, high blood pressure, increased risk of stroke and heart attack, and so on. If you have lots of these receptors, you may be able to eat lots of fat and maybe not have any real serious heart problems. It really just depends. And so here we see this example. Here's our LDL, our combination of protein and fat. Here's a membrane of our red blood cell with these receptor proteins. They bind that LDL. They're going to take it in through receptor-mediated endocytosis, digest it, and it's harmless. And so our receptors on our blood are directly linked to our genes. And again, that can certainly put you either at a higher or lower risk for something like heart disease. So to summarize today's information, we started off talking about endosymbiosis, that idea that eukaryotic cells came from prokaryotic cells that ate other prokaryotes, and those cells inside evolved into organelles like mitochondria and chloroplast. Then we certainly spent most of our time looking at cell transport, all those mechanisms that chemicals can use to cross the membrane, physically pass through the fats or the proteins, like diffusion, osmosis, facilitated diffusion, and active transport. And remember, diffusion, osmosis, and facilitated diffusion are all passive. Chemicals move from high to low. They're moving with the concentration gradient, with that natural flow. Active transport, chemicals are moving upstream against the gradient, and cells have to spend energy. Then we mentioned without crossing the membrane, chemicals can still enter or exit the cell. They enter the cell through endocytosis and exit through exocytosis. We talked about our different types of endocytosis, and in particular we talked about two important human disorders. We said cystic fibrosis, directly related to that combination of active transport and osmosis for water regulation, and then certainly high cholesterol and increased chance of heart disease associated with that receptor-mediated endocytosis.